Hi everyone. This is going to be the lecture for 8.11 to 8.14, and we'll be specifically focusing on meiosis. And today I wanted to try something a little bit different, so I'm going to use Prezi instead of PowerPoint. Okay, so just to give some background on chromosomes and some more technical terms, the regular body cell that we have, which is all of our normal body cells, are the ones that do mitosis, that process we've been discussing from before. These body cells are called somatic cells, this bolded term. For most of our body cells, all of our somatic cells, we have 46 chromosomes. That's equal to 23 pairs of chromosomes. So 46 chromosomes is how much we have total. Autosomes is a name for any chromosome that's not our sex chromosomes. And then sex chromosomes are special chromosomes that designate what gender you are. And our sex chromosomes are either XY for male or XX for female. If you look at this picture, this right here, this graph is a picture of all of our chromosomes in our body. So if you count, they've grouped the pairs together, but these are really 1, 2, 2, and then 4, and then 6. And so you notice at the very end we have 46 chromosomes total, 46 distinctive chromosomes. They put the two together because these are called homologous chromosomes. So they're a special pair of chromosomes. They're different from each other, but they are related in that if you look at them, they're very similar in size and shape. So not just so that you don't get confused, this is a duplicated chromosome. So these two sides are sister chromatids that are exactly the same. But this right here, this chromosome is a homologous chromosome to this one right here. And so these two homologous pairs, they have very similar size and shape, and they actually have matching genes. So if you look, it's color-coded. This darker green part would correlate with this darker green part on these two homologous chromosomes. And so they would be genes coding for the same kind of characteristic. So for example, this might code for, this right here might code for color of eyes. And so on on one chromosome, this right here could be green eyes, and then this one right here would be blue eyes. So it's the same characteristic, but you have different manifestations of that based on the genes. This could be like for curly hair, this could be for straight hair, but they both code for hair texture or hair shape. So homologous chromosomes, they tend to code for the same kind of characteristics, but they might have different genes for it. Let's see. So almost all of our chromosomes have matching homologous chromosomes. The only exception to that is the male sex chromosomes for X and Y. They tend not to have correlating, they tend to be just different from each other. Okay, so for somatic cells that have two sets of chromosomes, one set is inherited from each parent. Because we have those two sets, most animals and plants that have this are called diploid organisms. This word diploid just indicates that we really have double the number of chromosomes for each characteristic. So for us, we would have 2N, which is 46 total chromosomes, and each of those chromosomes has like a homologous pair. However, there's something called gametes. So as opposed to our somatic cells, which are regular body cells like skin cells and hair cells and all of those rest of those kinds of cells, gametes are sex cells. And in our sex cells, which is either your egg or your sperm cells, you only have one set of chromosomes. So because of that, they're called haploid cells, which means they only have one set. So N equals 23. They only have 23 chromosomes in these gamete cells. And whereas somatic cells are created by mitosis, where the parent cell just divides and they have genetically identical daughter cells, these gamete cells, which have half the number of chromosomes, are formed by a process called meiosis. This is just a process of cell division where it reduces the amount of chromosomes by half. And when you have these two sex cells that come together, a sperm and an egg cell, it's called fertilization. And then you get the 23 plus 23. It reforms a 46 chromosome cell. And then that's called a zygote, where you have one set of each homologous pair. Okay, so meiosis as an overview. This is the process, remember, that leads to your sex cells or your gametes, which are the haploid cells. In interphase, it's exactly the same as the interphase for your regular somatic cells. You have your normal chromosomes and they double, so they duplicate during the S phase. So now you're at the end of G2, you have double the number of chromosomes. So you have 46 chromosomes, but it's double the genetic material. In meiosis 1, it actually separates the homologous chromosomes, and you end up with two daughter cells that have half the number of chromosomes. So don't get confused, because you can see that they're still in their sister chromatid form. So right now this has kind of like double the amount of DNA, but it's double the same exact DNA. So this is still considered a haploid cell because it's missing half of its chromosomes. This one has the other half of the chromosomes, but it's doubled. And so in meiosis 2, you actually have each of these daughter cells divide into two more daughter cells. And now each of these, the thing is that all of them have the same amount of DNA as the original parent cell. So these ones, the sister chromatids just were pulled apart. And so you still have haploid cells here as equivalent, which are equivalent to this haploid cell. The DNA is just not duplicated. So that might be a little bit confusing, but we're going to go more in depth with that. Okay, so we're going to talk about meiosis 1 first. Okay, meiosis 1 is the first phase where we're taking the original parent cell and we're going to create two haploid daughter cells. Okay, prophase 1. Prophase 1 is the first part of this. So a lot of the normal things that happen in regular prophase for mitosis is similar. So you'll have the chromatin is condensing down to its chromosomes. And here, you don't really have a prometaphase 1 for meiosis. So they kind of put prometaphase and prophase together. So you have the nuclear envelope, which disintegrates in prophase 1. And then the spindle microtubules start to approach the chromosomes. The one thing that makes this prophase 1 different from regular prophase is there's something called synapsis, where the homologous chromosomes start to pair together. When the two homologous chromosomes come together, they form a tetrad. A tetrad is just when they align together. And as they align together, the chromatids start to overlap with each other. And as they overlap and cross over, they actually exchange segments of their DNA. So you can see in this picture, because they've crossed over, they actually swap some of their DNA. And then it leads to the two homologous chromosomes with different segments of DNA than their original form. And the reason for this is just to increase genetic diversity. So after prophase 1, you go to metaphase 1. All of these, now lining in the middle, are the homologous pairs of chromosomes. So these tetrads line up in the middle. And so the spindle fibers connect to the kinetochores in the centromeres of these homologous chromosomes. Then in anaphase 1, you actually pull apart these homologous chromosomes. So now as you're doing that, you're actually dividing the genetic material of each of these cells in half. Don't get confused, because it's double the amount of DNA, but the DNA here is the same. These sister chromatids, remember, are exactly the same as each other. This is different than this one. So these are homologous chromosomes. They're related, but they're not the same. And so you're taking away half of the homologous chromosomes, leading to two haploid daughter cells. Then at the end of telophase and cytokinesis 1, you end with two daughter cells that are now haploid. They have double the DNA, duplicated DNA, but it's now half of its original amount. Okay, so that is the end of meiosis 1. After meiosis 1, you have two daughter cells that are now going to divide again in meiosis 2. Meiosis 2 is going to be very similar to the previous processes. So now you have your two daughter cells, and what they're going to do in prophase 2, the nuclear membrane disintegrates, the spindle fibers start to form, and are going to start to connect the chromosomes. In metaphase 2, they're going to line up the chromosomes right in the middle of the cell, as they've been doing in the past. And here in anaphase 2, the sister chromatids are now going to be pulled apart. So you see the sister chromatids are going to be pulled apart, and you end up with four daughter cells. So four daughter cells from the two parent cells. Now these two parent cells, remember, were haploid, and now these 4 daughter cells are still haploid. They just have half of they don't have duplicated DNA anymore, but they're still haploid. Yeah, this is just to emphasize. Now, something that's different is that these 4 daughter cells are all a little bit different from each other because of that crossing over that happened where some of the chromosomes started to exchange DNA and have different sections of DNA from one another. You'll notice they try to show that here with the different colors, the red and the blue. But each of these 4 daughter cells are a little genetically different from one another. Okay, so your textbook has a really excellent diagram that compares mitosis with meiosis, and I would really take some time to look at it and make sure you understand like the differences and similarities. But you can see it generally is kind of follows the same pattern. Prophase, you start to see the DNA starting to condense down, and in prometaphase, which is included in prophase here, you have the spindle fibers that start to form, and the nuclear membrane breaks down. Metaphase, you'll have everything line up in the center. In mitosis, you just have all the 46 chromosomes line up in the middle, whereas in meiosis 1, the tetrads line up, so you have the pairs of chromosomes lining up, these tetrads, and so you'll have 23 pairs lining up, whereas here you had 46 chromosomes lining up. This leads, in anaphase, telophase, you end up with 2 daughter cells that are both diploid. They end up with the full 46 chromosomes. Here, at the end of meiosis 1, you have 2 haploid daughter cells that have double the amount of DNA. So these haploid daughter cells can divide one more time. And in meiosis 2, you end up with 4 genetically different haploid daughter cells. And right here, these are just kind of summaries that just summarize what we've been talking about. And just note that mitosis will be generally used for any cells that want to grow, tissue repair or maintenance of your cells, and then asexual reproduction. So that's what all your body cells normally do. Meiosis only happens for sexual reproduction when you want to form your egg cells or your sperm cells. I know that's a lot of material, so just make sure you understand all the processes, and after a while you'll realize that they're very similar. It's just that there's small differences, small details that make them a little bit different. Thanks so much for watching.